Hello. This is a video on the role the hormones play in the production of a plant meristem. Plant meristems are the micro-patterning source of the final large forms of mature plants. Meristem action is regulated hormonally by the impact of light and shade on the growing point. In the tip of a growing shoot, mother meristem is in the center of the meristematic tissue. Mother meristem is the site of cell division. The dividing cells reproduce horizontally. This action is stimulated by the light that produces the hormone cytokinin. Cytokinin is initially transported through the xylem from lateral roots, where it moves upwards to the meristem. In the mother meristem, it stimulates the hormonal growth of cuboidal cells. As the mother meristem cuboidal cells divide, they move outward from the center and down the sides of the meristem. There, the hormone Auxin is produced on the shaded sides of the growing meristem. Flowing downwards from the meristem, Auxin inhibits cytokinin. Auxin creates vertical, plane, apical domination by elongating the shoot tip up and away from the last leaf node. As a result, the apical bud always moves out towards a region of more light. Auxin acts to differentiate the dividing cuboidal cells in the mother meristem into narrow columns that stack up on the shaded sides of the meristem. Repeated layers of the columnar cells create internode tissue. As they proliferate, they elongate the stem, separating the leaf nodes in space. Apically dominant vertical growth patterns are polar to the nodal branch-forming potential of dormant lateral buds and leaf axils in the mother meristem itself. In the meristem, rhythmic waves of alternating but opposing growth patterns stretch and buckle the meristem to create leaves, nodes, and leaf axils, and the internodes. The nodes in the leaf axil form, and then will quickly become embedded in the rapidly growing leaf petiole tissue near the base of a developing leaf. The sequence of stem, node, and leaf then gives rise to the alternating pattern of the internodes between the leaf and the axillary nodal formations that will create branches in the future. The alternation of lateral and vertical growth is regulated by another hormone. Gibberellin regulates both auxin and cytokinin. It is a kind of a switching agent. There are times when cytokinin dominates. There are times when auxin dominates. Auxin with gibberellin dominates when the internode is extending, and cytokinin and gibberellin dominate when the meristem is creating new nodal conditions. So gibberellin acts kind of like a switch that regulates the dialogue between auxin and cytokinin. Gibberellin does this by lysing proteins to create enzymes that facilitate the switching response. Buds that are in leaf axils are released from their dormancy when xylem from the root comes up, flows through the petiole of a leaf, and then back out again into the cambium in the area between the leaf petiole and the bud itself. The cytokinin gets sequestered there and later on can act to have the bud shoot. The shooting of a new bud into a shoot here carries with it new leaves and axil bud, leaf axil bud, and that is generated by the cytokinin moving through. It is triggered by what are known as seasonal light signals. It's known as greenwood shooting. So this area here where the axil bud shoots out into a long shoot with its own leaves and nodes is known as greenwood. Gathering and fermenting different greenwood shoots at different times in the seasons with different seasons in the plant, especially say before fruiting or after fruiting, gathering the hormones that are in the greenwood shoot and then fermenting them in rainwater actually forms the basis of a rational system for making sprays to transform the greenwood hormones onto a cultivated plant. After a lateral bud is activated into a shoot, apical dominance develops from its own apical buds. This suppresses cytokinin availability to lateral buds further down on the shoot. Auxin gradually dominates the form through repeated internode lengthening of the lateral shoot by moving it towards the light. A repetition of this pattern leads to what is known as the approach to flower production. In this composite, we see the totality. Here's a branch that has had a shoot, and is this shoot here. And these are buds that are sequestered, that are not going to shoot out. Here is a leaf with a node. There's an inner node extending with a node in it. And up above in the approach, you can see that the inner nodes are getting shorter and the nodes are being carried up into the flower. So at that point, the hormones are balancing each other in very, very tight ways. The shoot compressing into the approach is a turn towards flower and fruit production. These kinds of patterns can reveal much that is mysterious in the year-to-year cycles of fruit trees. Sometimes they bear a lot, sometimes they don't bear too much. So to understand that, it's useful to understand that immature seeds, cytokinin dominates Auxin. That's what you see here. But if the fruits of an apple tree are left on too long, then eventually the cytokinin diminishes. And as the seeds mature, the cytokinin gives way to auxin. Abundant auxin in the mature seeds then generates something known as abscisic acid. Abscisic acid is a hormone that causes leaves to fall, but it also helps to mature fruit. And if the abscisic acid builds too much, then it's shunted off out of the fruit back towards the cambium. Abscisic acid then sequesters in the cambium at the end of the stalk from the fruit. This is all regulated by length of day signals. So the overripe fruit left on a tree shunts the abscisic acid into the cambium, but there it inhibits the fruit bud formation for the next year. At that point, spraying cytokinin-rich solutions in the fall from fermented greenwood on apple trees provides a sink of cytokinin that the leaf carries into the cambium at the base of next year's bud. This is utilized by the tree in the following spring to strengthen bud break and fruit production, since spring bud break is triggered by cytokinin. In nature, cold conditions for the bud over the winter gradually metabolizes the abscisic acid, allowing cytokinin in the cambium to influence bud break at the crank length of day in the next growing season. A caution here is that too much externally applied cytokinin in the fall can sometimes prevent phase change from a vegetative meristem to floral meristems. It prolongs the juvenile maturation state and doesn't allow the move to flowering. In general, auxin-dominated plants have very long midribs, like grasses, or very strong serrations. In general, cytokinin-dominated greenwood has very broad leaves with entire margins and no serrations. The choosing of different greenwoods at different times and fermenting them can be used as a rational approach to plant sprays, because if we pay attention, we can notice how the hormones are operating in a particular greenwood, ferment that in rainwater, and transfer those hormones to a cultivated crop. Thanks for watching.